Welcome to the Data Science Institute's virtual seminar series. My name is Dr. Sarah Mackey from Lawrence Livermore National Laboratory. Here at Lawrence Livermore, data science has become an essential discipline in many of our key program areas. LLNL is home to many challenging datasets, as well as home to some of the world's most advanced supercomputers. Our data science staff work in a variety of areas, including machine learning, artificial intelligence, big data analytics, statistical inference, predictive modeling, and uncertainty quantification. The Data Science Institute acts as a central hub for our lab's data science activities. We host events like this seminar series in order to introduce new ideas and potential collaborations to our laboratory staff. We invite speakers from outside the laboratory, from the Bay Area and beyond, to share their innovative approaches with our data science community. We're pleased now to include a wider audience in our seminar series through these recordings. You can read about past speakers at our website at data-science.llnl.gov, or you can email us at datascience@llnl.gov. Thank you and enjoy the seminar series.
We're now ready to introduce our speaker, Dr. Amanda Randles. Dr. Randles is an assistant professor of biomedical sciences and biomedical engineering at Duke University, focusing on the intersection of high performance computing, machine learning, and personalized medicine. Her group is developing new methods to aid in the diagnosis and treatment of diseases ranging from cardiovascular disease to cancer. She has received the NIH Pioneer Award, the NSF Career Award, the ACM Grace Hopper Award. She was named to the World Economic Forum Young Scientist list and the MIT Technology Review World's Top 35 Inventors under the Age of 35 list. She's a fellow of the National Academy of Inventors, and she received her PhD in applied physics from Harvard University as a DOE Computational Graduate Fellow and NSF Fellow. So it's our pleasure today to welcome you, Dr. Randles, and when you're ready, go ahead and take it away.
Great. Thank you so much. Okay, just double checking. You guys can see the slides from here.
Yeah, we can see them.
Great. So yeah, thank you all so much for joining, and I'm excited to be here. I'll kind of jump right in. I wanted to start off and just kind of give you some orientation of what I'm excited about, what I'm thinking about, what I'm hoping we can convey in this hour. So basically, I'm really excited about developing what we're calling vascular digital twins, in the hope that we can improve physicians' ability to find disease, track it better, and ultimately improve our ability to treat human diseases. And I'm, of course, really excited about, how do we use large-scale computation? Often, how do we use, some of the world's biggest supercomputers like you have at Livermore to really help this cause and move this forward? So everything I'm working on is really driven from the biomedical question and the impact that we can make on the biomedical side. And then we're trying to develop the computational tools and those capabilities as necessary to really achieve our different goals as we kind of move through these biomedical problems. I'm excited to talk to you all because I think the work that we're doing creates a lot of data, and there are a lot of opportunities for novel, interesting application of data science in this space. And I'm hoping today through the talk to kind of just highlight a lot of different areas where I think data science can make a difference. I'll start off and say I don't know if I would classify myself as a pure data scientist. I think I'm definitely more on the application side, but I'm excited to talk to a group like this and kind of see, if we show you what we're working on, where we can kind of come in the middle, and if there are opportunities for collaboration or spaces where the kinds of things you guys are working on, where we might see that making an impact on the biomedical space and kind of coming from that avenue. So just jumping in, I think there are different ways that we can really use these types of digital twins. In our lab, we're really focused on primarily three main areas, and that's what I'm going to talk about for the rest of the hour. We're trying to use these personalized models to improve our ability to diagnose disease, and to do so in a non-invasive way. This also allows us to have the capability of improving and providing a way for the physicians to do treatment planning. So I'll delve into what I mean by that a bit more later on. But think of essentially you have this 3D model of the patient. So before they ever step into the operating room, we're giving them that opportunity to decide, what stent should I place? Where should I place it? What would be the impact of putting, the stent at a slightly different angle or a different shunt at a different angle? Or how do you really, try these out, and things you could never really try out in the patient, but do so virtually and see an accurate and efficient view of what's going to happen and how these different treatment options will affect the hemodynamics, the blood flow, the outcomes for those patients, and allow doctors to make more informed decisions and improve their ability to plan and identify optimal treatment in the future. On the other side, I think everything on the top is much more translational, much closer to, how do we actually get that into the clinic today? But we can also use these models to really improve our underlying biological understanding. So we're also trying to focus more on the cell-based side to get at what are these mechanisms driving cell transport, driving disease localization, why is it progressing in the way that it is? The use case and example we tend to look at is cancer metastasis and trying to understand, once it's a tumor, once a cancer cell moves off the tumor, goes into your bloodstream, why is it going to go, why is it going to go to that secondary site? What is it about that secondary site that's causing it to spend more time there? And why are you getting sites of metastasis where you are? So for the rest of the talk, we'll kind of frame it in these and kind of going through these different goals. But first, I kind of wanted to pause and just make sure we're all on the same page and you understand what I mean and what I'm talking about when I'm referring to these vascular digital twins. So the way we're building these, we're taking patient-specific data. So we're taking conventional scans. It can be an MRI scan, a CT scan. We have tools to take two different two-dimensional images and reconstruct the 3D geometry. We're personally using commercial software. So we're not focused on the data segmentation piece, but that in itself is an entirely different, many PhD theses and a lot of work. And I'm sure from the data science side, people may have seen this before of trying to understand how do you improve data segmentation. But mainly from our end, we want to identify what is the anatomy of that specific patient and how do we pull out a 3D mesh file representing that patient's anatomy. We're then applying a regular Cartesian grid to this 3D mesh file. In this case, you're seeing coronary arteries. We apply this regular Cartesian grid, and then we have to determine what's an inlet node, what's an outlet node, what's inside, what's outside the mesh. And then we're solving fluid dynamics equations at each of these grid points. In our work, which I won't really delve into the math at this stage, but if definitely pause and ask questions if you're interested. We're using Lattice-Boltzmann, which is an alternative to the Navier-Stokes. If you're familiar with fluid dynamics, typically people will use the Navier-Stokes equations. You can recover the same fluid mechanics from the Lattice-Boltzmann approach. We're just taking a discretized view of the fluid and a discretized view of the velocity space. It allows us to use this model that lends itself extremely well to parallelization. And that's where we end up needing these large-scale supercomputers and why we've been doing a lot of work with Livermore over the last 10 years to really scale these codes and allow us to get extremely high resolution, extremely fast models in these very complex geometries that you see in someone's vasculature. In this talk, I won't really delve into our validation and the biomedical side, but I think it's always important when you're seeing these talks, there should be some comment on why should you actually believe that our simulation results are accurate. So I kind of threw in the feedback loop of, we have worked very closely with experimentalists over the last decade or so to validate our code and ensure that we're getting the right results. I can answer questions on that if people are interested later. If we've done a lot of work with 3D printing, we've done a lot of work with the teams at Livermore that are doing 3D bioprinting to really validate that we are getting, using particle image velocimetry, the right flow patterns in these complex geometries. Because we're moving away from the geometries where you have an analytical solution, and we're trying to ensure that we're able to get flow parameters like velocity, pressure, wall shear stress, accurate in these high resolution simulations. I will touch on it in a bit, but we also have completed a large clinical trial where we are comparing our results to invasive measurements where they're putting the guide wire in the patient, directly measuring the pressure in the patient, and we're able to compare that to our simulations as well. So we're always thinking about, how do you make these simulations more accurate and what goes into these models to ensure that they're as accurate as possible. And we also want to put them back into the clinic. So we're also thinking about, how do we make this usable by the doctors in intuitive by the doctors. We've actually spent a lot of time looking at things, down to, what's the best color scheme to use to get the doctors to be able to find regions of likely disease localization faster, more accurately. And then when we're doing the treatment planning, how do we provide that data back to the doctors in a way that is intuitive and easy to interact with and, mimic what they're going to see in the clinic to be able to improve their ability to complete virtual surgery planning in the future.
Amanda, before you move on, we've got a question from Mark Miller. He's wondering about the resolution of the vascular system scans and what resolution you can achieve without injury to the patient.
So I will say, and I think most of you are more comfortable in the computational side. You can kind of appreciate from the computational end. From our end, going into the simulation, we just need the mesh. We are really gated by the resolution you can get from the imaging capability. I'll show later, we have one model where we've looked at the systemic scale, and that's down to about a 1 millimeter in diameter resolution, but we can go further depending on that imaging modality. For large scale scans, we were trying to piece together, and it's usually not one scan, it's usually trying to piece together different passes. We're usually limited to about 1 millimeter diameter, but we can go further if you're using like angiograms where you have a conventional angiogram. For some studies, when we're looking at other work, outside of the body, we might get into like micro CT. We've actually been talking to people about using photoacoustic imaging for, you can't, the depth is more of a problem, but you can get high resolution vasculature. So it's completely gated by the imaging side. So that's where, we are working with a lot of imaging researchers trying to figure out like how do we push the bounds. And then from the computational end, we want that CT, we want the triangulated mesh. And then to kind of show what we do with this mesh and kind of just give you that intuition and the picture in your head for what we're working on. I've been showing this video. If you've ever seen a talk, I think I've shown this video since 2010. Hopefully we'll replace it soon. But it does give you the idea and kind of more of an intuition of what do we mean by these vascular twins and why are they actually so computationally intense. So the first half of this talk, I'll really focus on bulk fluid simulations, but the second half I'm going to focus on what can we do when we actually resolve individual cells. And this model was run back in 2010, but on one of the Argon systems that had about 140,000 processors at the time, which in 2010 was the biggest supercomputer at the time. And just for scale, it took about 6 hours to run, or it took 6 hours to simulate one human heartbeat on that scale, including about 200 million, 300 million individual red blood cells. But this also gets at why this data, why these models end up being so computationally intense. Because you're trying to model the individual red blood cells, their interaction with the fluid, with the wall, etc. And it kind of ends up being why we need to work with teams like those at Livermore to get these kinds of large scale simulations. So kind of just summarizing, when we're looking and building these models, it touches on a lot of pieces of computer science, computational science, data science. We run into many issues just trying to create, even just those visualizations are incredibly computationally intensive and data intensive, where we care about anything from the domain decomposition and how do we actually scale on the large scale supercomputer to how do we visualize it, how do we interact with it, how do we actually, what do we, now that we have all the data, what can we do with it? And so there's a good question in the chat on, does the blood flow at 1 millimeter resolution need to be modeled down to individual cells? And I think that's perfect timing in going through the two different sections here. You don't always need all of the cells. It really is guided by what question you need or what question you're asking. So any of our work where we're trying to understand, like when you go back to this model, the grand challenge we're working on now is imagine dropping in a cancer cell and understanding the interaction of the cancer cell with all the red blood cells that are causing it to go down different paths. Where is it going? How is it spending time? And what is really causing it to spend, increase its residence time at different locations? How can we predict where it's going to go and understand, the basic understanding of mechanisms driving cancer metastasis? On the flip side, if you're looking at cardiovascular work, in many cases, you don't actually need to go down to the level of the red blood cells. We can get away with much coarser resolution grids. And when you're looking at when you're looking at the bulk fluid simulations, we can actually increase that resolution or the scale significantly. So it really depends on the question that we're asking. So that comes back to, we'll start here of how do we actually use these models? And I'm going to try to point out some of the data science questions as we go through, but motivated from the biology. So in our work, we care a lot about, the way we're seeing these computational models first get into the clinic has really been on the diagnostic side. And I'll walk through one use case, but we're already seeing FDA approved tools. We're seeing them being adopted in the clinic. And this is really, the first real forefront for these kinds of computational models. And the next step is really transitioning into treatment planning and then moving to other regions of the body. So there's a lot of work in the coronary and the cardiovascular space. In that work, we need to, it's always important. Real-time simulation is always important. We want to, overall, even if you can't get to real time, you want to decrease that time to solution all the time. We need to provide this intuitive means to interact. But then we also want to increase the scale of the domain that's being modeled from just those coronary arteries that you just saw in our other simulation to much larger regions of the body and move away from just focusing on the coronary to can we look at, something on the scale of the full body and understand global effects of different treatment options. And then we also want to look at, can we move away from a single heartbeat now with your geometry as it is today to what happens with different treatment options. So the first use case I wanted to focus on is I would say this is probably the canonical how are we getting non-invasive models into the clinic. It's looking at coronary flow. We're looking at cardiovascular disease where you have a lesion in your coronary arteries. So what you're seeing with this yellow is really the plaque buildup, and that's what we mean by a lesion, like that narrowing in the coronary artery that's really influencing the blood flow through the heart and leading to chest pain, leading to these adverse effects. And what the doctors need to do is determine should they, for some reason it starts going through, should they or should they not stent this lesion. And the way they currently do it in the current standard of practice is they take an angiogram, they determine where the lesion is, and this is an angiogram image that you're seeing on the side. They will then place the guide wire in the coronary artery and measure the pressure, and that's where you see the PD. They'll measure the pressure before the stent and after the stent, or before the lesion and after the lesion, take the ratio of those two pressures, and if it's above or below 0.8, they will determine if they're going to place a stent. And it's purely, if it's above or below 0.8, they will decide that's determining, are they going to intervene and place a stent for this patient or not. Digital twins offer the capability of taking a patient-specific simulation, creating that 3D model of the patient's coronary arteries, running the flow profile, and then you can measure that pressure gradient in your simulation without ever having to use that guide wire. And we're moving away from an invasive measurement and using these non-invasive measures. Here, we've recently completed a 200-patient trial. We did this with between Brigham and Women's Hospital and Duke University Hospital here. We have, I don't know why. We'll come back. Okay, so we've completed a 200-patient trial where we have the measured invasive guide, what they were getting for the measured invasive guide wire, and shown that you can use our tool, which is called Harvey, to accurately model and calculate this what's called fractional flow reserve, but this pressure gradient essentially. It's interesting, this is a great first step, and this is why you're seeing tools like this are currently FDA approved. We're getting an average error of 6.45%, and we're able to actually really replicate very accurately that pressure gradient. I think what's most more interesting and a little bit more exciting is that we can actually go beyond these conventional metrics of the pressure gradient. And we've identified that a metric like vorticity, so where you're seeing this disturbed flow, the patients who are right at that borderline, so if they're at, maybe at 0.81 or 0.805 or right above that 0.8 cutoff, that wouldn't have normally been treated. If they're in what we call the gray zone, so right at this cutoff point, if they have an elevated vorticity, we actually found that they were more likely to have adverse effects down the line, indicating that aside from FFR, this pressure gradient, we may be able to use something more complex like the vorticity Sorry for the slides automatically moving, like the vorticity to determine if they should or should not receive the stent. And examples like this are the first pass of like, how can we use data science to really identify these other metrics, these more complex metrics that you can really only get out of these like high resolution 3D flow simulations and improve treatment planning? Can we provide other metrics and other options for the physicians to kind of augment these metrics like fractional flow reserve? The next step is it actually allows you to capture more complex lesions. So typically when you're looking at these clinical trials, they'll actually leave out these types of patients because they're more complex lesions that aren't as conventional, but complex lesions can be found in more than 20% of these patients, and they have a higher risk of having these adverse events. So by a complex lesion, we mean things like at the ostium or a serial lesion. So where you may have, one lesion immediately followed by the next lesion. So trying to determine, do you need to stent both of these lesions? Do you need to treat just one of them? And what is it like, how do you actually treat these is a key question for the doctors, but also how do we identify them? How do we characterize them? How do we diagnose them initially? Because they're much harder to determine. We're focusing a lot on bifurcation lesions in our lab right now. And a bifurcation lesion are the ones where they're at, we have here, we're calling it the main vessel and the side branch, but you can also kind of see, you could have a lesion at any of these points in that wide bifurcation. So doctors need to know before they go into the operating room, do I need to stent anything in just this main vessel? Do I need to stent both the side branch and the main vessel? Like, what is the best option for this patient? Which stent should I have? What stent should I have? Like, what should I grab? Do I get a kissing stent? Do I get a single stent? Like, what is that right treatment option before they ever start the surgery? They obviously can't try, wouldn't it be great if I can just stent the main branch, see what happens, and then determine, based on the results of that, should I then stent the side branch? You don't really want to do that in the patient itself. So we're doing a lot of work in collaboration with Brigham and Women's Hospital and Rice University to figure out ways of classifying these patients to see, can we give a classification system to determine when you should be treating the main branch and the side branch, or just the main branch, and provide more feedback and guidance on how to treat these types of lesions. What's great about the physics-based simulations is we can, one, virtually treat the patients. So you can take the geometry where you have like this lesion here, right at that bifurcation. You can modify it so that you do actually, make it look like you treated that main branch or that main vessel, and see what would happen for those patients. What was the flow like before you treated the main branch? What was the flow like after you treated the main branch? And use that to determine, what was the FFR remaining in the side branch after I treated the main branch? Should I really be treating the side branch? And do all of that virtually. Moreover, we can use these simulations to create a large synthetic database. That's where a lot of the opportunities, I think, coming into the data science side of it are really at. We can create targeted synthetic databases that are really controlling everything in the geometry except for quantities of interest in a way you just couldn't do in the clinic. If you were going to create, 1,000 patient data set, it wouldn't just be, that the length of the lesion, the length of the lesion would be different. They would have a different number of branches in their coronary arteries. There would be different, different angles, different diameters. There are many things that differ between each patient. And by creating an intentional and targeted synthetic data set, we're able to take many different base cases to capture different patients, but then systematically change the curvature. And here we're showing like 3 key metrics of, the curvature, if it's smooth or is it sharp, the length of the lesion, the severity of the lesion, and really tease out what is the impact of each of these components and how does that, what are the synergistic effects with different metrics that actually describe the patient's anatomy itself. So this is our very first, basic side, and I would say it's almost embarrassing to present this to a group of data scientists. We're starting with the goal of trying to getting a method, we want to have a decision tree and classification system we can hand to the doctors that they can understand. So we're starting with very simple and really basic data science methods. So here's just a cart model. But it's providing an interpretable method that allows us to create this decision tree. And this is showing the main branch at the top, the side branch on the left. And what's great is you can it identifies what are the influential anatomical features. So the bifurcation angle ended up being incredibly important. The number of side branches in the coronary arteries ends up being very important. And it allows you to create this systematic decision tree. In this case, we're looking at wall shear stress, which is actually a quantity known to be associated with progression of cardiovascular disease. But it allows us to create, if you're looking at, predicting wall shear stress within the main branch, it gives you this decision tree that essentially says, if bifurcation angle is less than 36, this is like your likely time average wall shear stress. If your bifurcation angle is then, between 36 and 43, and then how does that grow with the side branches? And we're able to start taking into account a lot of these other, characteristics that are much more complex into one model. And in this case, if we can build enough of the synthetic database and build this into the system, we wouldn't even need to run a simulation for a new patient. You're just going to follow this type of decision tree and use these types of regression models to fit and identify, is this patient likely going to have a fractional flow reserve or a pressure gradient in, a region above or below 0.8? Can we get, what is the uncertainty associated with each of these predictions? And can we use this as a first pass model to either determine, if this says their fractional flow reserve is going to be, 0.6 or far enough away from the 0.8, maybe that's good enough. Maybe it's indicating which patients you actually do need to go in and run a 3D full simulation to really check if they're in the gray zone. When can you use a reduced order model, and how can we use these kind of combinations to start creating classification systems to feed back to the doctors to build on that. The next step is bringing it to, kind of building off this side branch and main branch idea. Can we build it into, in the bifurcation, it's very dichotomous of, have we treated the main branch? Have we not treated the main branch? What is the effect of treating that main branch? In reality, we want this to be much more pervasive and say, can we move away from just classification to prospective prediction of saying, for this specific patient, here we're looking at coarctation of the aorta. They have, again, this narrowing that's causing a huge pressure gradient that really is the issue, that once it's above 20 millimeters of mercury, the doctors are trying to treat. They could treat this with anastomosis. Ooh, it looks like it's okay. But you can treat this with anastomosis, where you're removing that lesion. You can put a balloon in and do angioplasty. You can place a stent. There are many different options. And can we virtually simulate each of these different options and determine, what is best for that specific patient? Can we also try, different types of stents and seeing, which stent is best for that patient, which location is best for that treatment? You can kind of imagine, many different virtual interventions that we want to test out for those patients. For pediatric cases, we're putting shunts in to get blood flow to different points of the body. You can imagine different angles. We might look at an hourglass shape. We're trying to optimize for these different patients. What are the different ways of doing this? And everything I've talked about so far is for the coronaries. How do we actually apply this to the legs and other regions of the body where you actually do have similar cases where you have these large, you'll have a narrowing, and how do we move that away? This kind of comes back to, how do we actually make this feasible? And the first step in moving from the coronary model that we had, that I showed you from our 2010 paper, to moving to a much larger region of the body, the first and really necessary step was minimizing our overall memory. We did this work that I'll touch on briefly to just get from the coronaries and move to the level of the full system back in 2015. So that was back on Sequoia at Livermore. And the things to remember here, the entire system at that stage had 1.6 petabytes of memory. So memory and trying to optimize your memory usage was a critical and necessary first step. Even now on the newer systems, it's, maybe 5, 10 petabytes. We're not getting to, if you have this, the 9 micron side, would have needed 90 petabytes of data from a naive implementation. That's really just not feasible. So kind of the first step was, it's necessary to have semi-direct, indirect addressing. I won't kind of go into the deep end of all the different things we did to optimize this, but again, happy to talk about it if you're interested. We've tried things like, instead of saving the velocity space, we've moved to the moment space, and that actually saved 47% of memory. We've looked into different memory addressing schemes, so thinking of structure of arrays versus array of structure, and how do you map that to the GPU versus the CPU as you're going to different systems. Initialization and load balance ends up being incredibly important for these models. Again, I won't go into all the details, but it's, you start having so many grid points. Like here, we have at a 20 micron resolution, we already have 45 billion grid points. Having a conventional neighbor list of where are these grid points going to be located isn't really feasible and tractable anymore, because just that would take all of the memory on each compute node, let alone your entire simulation. So just the key point here is you have to start thinking of, how do you actually move this to these large scale systems? You start running into problems you might not have at 100 processors or 200 processors. That being said, we've done a lot of work to kind of optimize the Harvey simulation. We've worked with a team at Livermore to do this. And the key takeaway is we're actually able to now run on these large scale systems. At that point, it was Sequoia with the 1.5, 1.6 million cores. And we can do so efficiently. From the data science side, that's exciting because that means we can actually, we are making use of that entire system, and we are generating a ton of data for all of these models. And I'll kind of touch on how we actually use this and what we can do. So this shows just the impact of being able to move from, 140,000 cores to getting to about 1.6 million. And what we had here was from that video from 2010 with just doing the coronary. And this is a simulation showing, what we were able to do moving to the full body then in 2015. So we're able to move, from like small scale to getting to the much larger. From an interaction standpoint, this is great because it allows us to actually start doing virtual surgery and able to show that using these simulations, you can see the changes on a local level. So here I'm looking again at this coarctation of the aorta. We're modifying that coarctation, and we're seeing a tangible impact on what we call ABI. And this is your ankle brachial index. So it's essentially, again, that ratio, but it's a ratio of what the pressure is at your ankle versus your arm. And they use this a lot to determine the severity of peripheral vascular disease. So they'll use this when you have lesions. Typically, they'll actually use it for many lesions in the leg and understanding, what is happening on a global scale for that patient. And we're demonstrating here, and you can just see that the ABI significantly changes if you have like an 80% lesion versus, serial lesions that are 60% lesions versus what the patient originally had. And the key step is we're really trying to keep making, little steps in how do we actually get virtual surgery in, be able to actually represent virtual surgery and see this treatment planning for these patients. As you can probably appreciate, the big issue here is that it is a huge computational and memory burden. We don't want to have to run a new simulation that requires all of the world's largest supercomputer for every single treatment option that doctor needs to try. That's really, not tractable going forward. And we're creating tons and tons of data, and we have to be able to handle that side as well. So I wanted to go through a few vignettes in the surgical planning space and the treatment planning space of how are we trying to deal with this. The first one is what you may be obvious and come up with is, couple this with reduced order models. Kind of when you were asking about, do we really need the red blood cells in each case? We don't even need a 3D model in all cases. So how do we actually efficiently couple 1D models with 3D models and use this when we need to? One example was looking at VA ECMO. So this is a mechanical support system where you have your heart or your lungs are really starting to fail, and we're pumping the blood outside the body, and outside the body you're oxygenating it and then infusing it back through a cannula into the body. The thing I wanted to point out here is when we have that insertion cannula, there are many different places, there are 3 different places they can reinsert that cannula. They have the option of the femoral artery, the axillary artery, or the aorta. So using these simulations, we can determine where should you place that cannula, what's best for that specific patient, and then how do you actually set up the ECMO device to optimize for that patient. One of the things they want to look at is the idea of this mixing zone. And as you watch this video, you'll kind of see there's blood flow coming from the heart as it's starting to recover, and there's blood flow coming up from the bottom, and that's coming from the machine, depending where you're cannulating and putting the flow back in. So you're having these 2 streams of flow that are actually coming together and mixing and hitting. And what you see there is defined as that mixing zone of where they actually hit. The location of that mixing zone strongly influences how much oxygen is going to get to the rest of the body, get to the brain. And depending on how that flow is happening and what the flow rates are, especially coming in from the ECMO system, that can lead to lack of oxygen in the brain, which is going to lead to hypoxia in the brain and neurological complications. 15% of patients on ECMO end up having neurological complications. So really understanding the influence of even just like, the location of the cannula, but also things like the flow rate and the different settings that the doctors can change on that ECMO system, we can help tune it and improve it on a patient-by-patient basis. In this setting, it's very important to understand the mixing zone location. And if what you really care about is the oxygenation in the cerebral vasculature and understanding how much oxygen is getting to the brain, the 3D flow of the brain to capture that oxygenation level can be very important, but you don't necessarily need a 3D model of the rest of the body. So we've introduced methods to couple that 3D model to the 1D model and put that together. And that allows you to assess the effect, and here is showing the effect of different cannulation and the velocity that you're seeing from the ECMO device. So the velocity of the flow that was coming up from the bottom, whether you're cannulating in the leg or you're cannulating at the axillary or at the central, meaning at the aorta and the heart. And it allows you to help determine what is the effect of the flow rate of the system, how should the doctor be setting that dial, and where should they be cannulating for that patient. So it's really a first step in trying to understand how do we guide treatment and how do we inform treatment planning for specific patients based on the question. Aside from coupling reduced order models, we've also started looking at questions like, our first foray into machine learning was really more of a design of experiment approach of using machine learning. So coming back to that coarctation question, it's a congenital heart disease that affects maybe 3,000 to 5,000 patients in the US each year. The issue is they will intervene if this pressure gradient gets over 20 millimeters of mercury. We also really care about wall shear stress. In this case, we're looking at time average wall shear stress. That's associated with plaque progression. We want to be able to understand what is the pressure gradient in this region for a specific patient and what is their potential wall shear stress. From the data science side, and maybe a more interesting data question, the key point we started looking at was, right now you look at the pressure gradient for that patient when they're in the office and for the doctor. You take that one pressure gradient, and then you make all your clinical decisions based on that. And that's not taking into account what could happen to that patient as they go about the rest of their life. What if they are, exercising heavily? If they're running, what if they're running in Denver and you're at high altitude? How does that affect the patient? And what happens if, they're perfectly fine unless they start going running in Denver, or if they're, third trimester pregnant running in Denver? And how do these things kind of couple with each other? And how do you take that into account? The brute force is really trying to create this holistic view of the patient by actually simulating each of these different models. We actually did this to get our ground truth. Again, that's not really tractable as you move to many other patients, because you're not going to be able to do these large scale simulations on big supercomputers and try to get, essentially this heat map that you're seeing here as you change the flow rate, which is going to capture, differences in exercise level for that patient as we go along, viscosity, which can be influenced by things like like your hematocrit levels. So if you're anemic, if you're pregnant, that may change. If you're at altitude, so all the different things that may change your blood flow and try to get this heat map. It's really not tractable to get this heat map by running all of these individual points. But the doctors are starting to move from trying to have the single view of the patient to getting this holistic view, and how do we actually help them capture that? So that's where we started using this design of experiment approach using machine learning. And the goal there was really to narrow down the number of simulations you actually had to run to effectively treat a machine learning model to predict what, in this case, we were looking at pressure gradient and time average wall shear stress. We were able to identify you can actually train a minimal model with very few simulations, and we identified what the pairings were, so which simulations you needed to run. So the pairing of the flow rate with the viscosity, run those specific simulations, identify which ones needed to be run, the minimal number that needed to be run, and that way when a new patient came in, you actually only needed to run those few simulations to be able to train a model and predict that entire state space for each of these patients, and therefore drastically reduce your overall simulation time. So kind of paths like that are ways that we see of kind of bringing in this is a very basic, first step. How do we kind of build on that and that kind of data to improve our ability to predict and get a much more holistic view of that patient and how they may go about their daily lives and risk factors as they, when they're away from the clinic. Kind of coming back to that initial point that I had mentioned on the large data, I think it's key and important. I think you guys all may have many other ideas. So we can, when we're looking at the full body, the question is like, do you model the effect of gravity and blood flow? And we do for the full body. We can actually do the tilt tests where you like rotate the model and add in the effect of gravity. We don't need that for many of the studies, but when you get to the full body scale, you actually do have to take that into account. And I wanted to point out here, we do kind of the third vignette in this space is trying to, further our use of machine learning. But yes, yeah, I agree. Yeah, sorry, it'd be useful for space travel. It could be really interesting. And then we want to be able to predict, aside from these different state spaces of are they running, are they in Denver, all of this. What happens for these different treatment options? Can we actually provide more interactive, immediate real-time feedback of if I place a stent here, how does that affect the flow? We don't want to place a stent and then have a 4-hour simulation that has to go and run, because then the patient may have left the clinic, they may need to go re-image, they may, that adds in if they want to try 10 different options, have to wait for 10 different 4-hour simulations. How do we actually build real-time models? And a lot of that is going to come back to how do we leverage machine learning? How do we do this intelligently, aside from the reduced order model coupling? And with that, I wanted to point out that, while we are only running, even in this one simulation, it's one patient, which is very, counterintuitive to how most people, when they're looking at these large patient trials and you want to use machine learning, you want to do it with millions and millions of patients. We're actually generating a ton of data. And so I think from that side, it's always worth coming back when we're running a 20 micron simulation or a 9 micron simulation, like you're generating tons and tons of data, but you're also generating, and that it'd be more interesting, I should put the square down below. Even in the optimized case where we're not creating the entire bounding box, we still have 509 billion fluid points that we are simulating in the 9 micron simulation, and we're simulating that for every single time step. And typically that is a big trade-off of lattice Boltzmann is we have really, really tiny time steps. So we end up modeling millions and millions of time steps, and we're running, we're getting that data for 509 billion fluid points. That's giving you a lot of data to be able to train these models and try to create more real-time interaction. What can we do to say, if I were to modify the geometry in a certain way, can I infer what the simulation result would be using any of these other machine learning models to augment these simulations? Can we have some surrogates to try to predict how would that impact be, and do that in a more real-time, more interactive model? I think one of the key examples here is coming back to that coronary space. We want that real-time interaction. Currently, there are FDA-approved tools that have real-time interaction, but the caveat here is they have to train for a whole day for each new patient before you get that real-time interaction. So we're trying to develop models that actually reduce the overall training cost, even if it does provide you have that real-time interaction, because in the clinic, you really do want to have the training. If you can limit the training time down to like 10 minutes, that changes the game for the doctors. They may, if they need to go back and get more images, that patient may still be there. If they need to interact and do this, get information quickly, you can imagine cutting it from a day required first down to 10 minutes can have significant impacts on the health side. And then we also think about how do we make it interactive and how do we make it intuitive. And I won't kind of go on too much of a tangent here, but I think it's something, we're always trying to figure out how do we get this in the clinic in a way, the doctors don't know C++ programming, they don't know command line programming, they don't know how to, work with Slurm and set up the, large scale simulation tools. So how do we actually give that to the doctors in a way that they can place it? So we've been working a lot with developing VR, extended reality, interactive immersive tools to understand when is immersion important and how does it help. And I won't go into deep dive here, but we have done a lot of quantitative user studies trying to just understand, when is this just a kitschy interactive tool, and when is it actually useful, and why do we want to see, when is it helpful for the doctors to see it in 3D space? Is it useful to have, you can kind of see in these images, here we're showing like the Z space, which is more of a semi-immersive where you're wearing the 3D goggles, and you have it like, kind of seeing it on the screen, versus having something that's fully immersive, like what you would have with an Oculus Quest or I guess Meta Quest now. But when what you would have with a Quest or you would have with a Z space, and how do we really do that in an interactive way? And I want to jump over to the cells before I run too much. And there's a lot of interesting questions if you take the other side of things, and how do we look at fluid structure interaction? So everything I've talked about up to this point is bulk fluid, very close to being in the clinic. How do we and all of those projects we're working on closely with the doctors. On the other side, you have the fluid structure interaction, and that's where we want to understand, how are the cells interacting with each other? Why are they interacting with each other in this way? And it provides an amazing opportunity from the computational end, because you have this controlled experiment where everything is exactly the same, and you can, go in and say, if my diameter of the cell is just slightly bigger, what does that do to its behavior? If the viscosity is slightly different, if it's slightly more deformable, if we change the mechanical properties of the cell, it gives you this capability that you could never control it to that degree in an experiment, in a wet lab experiment. So having this capability of doing it on the computational side is very unique and allows us to create large data sets in a way that you couldn't otherwise get to this kinds of data. And our goal here is, again, to model, we want to increase the temporal domain, so how many seconds or minutes you can simulate, but also pushing the bounds of what we can model spatially. And in both of those, that ends up creating a lot of opportunity for the role of data science. I want to just start with the motivation and kind of mention, different things we've done and why you actually should care about the cell mechanics. I'll go over this very briefly, but I think this one's kind of fun just because it was in collaboration with the team at Livermore. We worked with the team with Monica Moya's team and Rick Hines. We were trying to 3D bioprint these geometries, their endothelialized channels, and we're trying to couple them and create a digital twin of these bioprinted vessels to get at metrics that you couldn't measure in that bioprint. And see, here we're showing wall shear stress, which would be very difficult to measure in the bioprint, but we can show you what the shear stress is, and we were able to understand the impact that wall shear stress had on locations where they saw cancer cells adhering. So we can really kind of couple these more complex metrics with what you're seeing from the experimental data. And I think it's this coupling of the in vitro models with the with these digital twins that are really potentially powerful and show us where we can go. We have a lot of work going now with Lydia Sohn's lab up at Berkeley, and it's this similar idea of, how do we take this high throughput cell phenotyping technology and couple it with a digital twin so we can provide more data. So we have the data that we're able to measure with her phenotypers, and they're doing high throughput mechanophenotyping, but they can't measure things in a high throughput way like Young's modulus. And then using the digital twin, you're able to get out that kind of information very easily. So we're kind of looking for, how do we actually couple these experimental data sets with these digital twins. The quick overview of what we're looking at: we have that lattice-Boltzmann fluid solver coupled with a finite element solver for the red blood cells. You can kind of see on here that it's actually, it's an infinitesimally small triangulated mesh represented the red blood cells, the cancer cells, and they're connecting via immersed boundary. So it's really just like this exchange between the fluid nodes and our the triangulated models. I won't go into the deep end of the math there either, but it's really basically I think the thing you can appreciate here is we'll need a much higher resolution to actually capture that deformation of the cells. So it ends up being even more computationally intense as we move forward. Our first step and the first piece of adding in cells to your model is making sure you're actually adding one cell accurately. So we spent a lot of time trying to ensure that we were modeling red blood cells accurately and cancer cells accurately. So here it's focused on just one cell, but it's showing that we can match. We looked at micropipette aspiration and optical tweezers for the red blood cell, and we can tune our model to specific cell lines. So the blue is showing TMAT, which is one type of cancer. We have the cancer cell line. We have the L1210, which is another type of cancer cell line, and the black dots are showing what we're getting out of our simulation. And the key point here is we can actually tune tune these somewhat simplified models of the cell to specific types of cells. With that, we wanted to add in more interesting dynamics. So we actually went even further down in scale. So we're looking at cellular adhesive reactions. So we have, these are like the little bars you're seeing are individual ligand receptor bonds that are forming between the endothelium and the cancer cell itself. This is interesting because if you take into account the adhesive bonds, that can actually change the behavior and the trajectory of the cells. So it's really showing like why we need to go down to those levels and why it's important as we're trying to understand cell trajectory that we capture things on, like, capture behavior on that level. From our end, the next piece was then how do we actually, it's great, we can do one model, how do we move to many? So we spent a lot of time and effort scaling this up to over 500 million red blood cells in many different cancer cells. I won't spend a lot of time because I want to make sure there's a couple seconds for questions, but we've also parallelized the adhesion model. This is kind of showing our breakdown of, what is parallelized with the OpenMP and MPI side, what's only, anything that's just going to be handled on the serial component. And the key was this allowed us to move to modeling up to 160. In this case, we've demonstrated up to 160 adhesive cells in a complex geometry. It's kind of fun to see, like we're seeing this complex area. You can get down to the single cell adhesion. You can see the little dots on the cells are like each of the different receptors, and you can see the cells kind of adhering and moving through the geometry. So we're able to capture this kind of complexity, and that's great. But I think in this kind of, I'm trying to get to what I think you guys might find much more interesting. We've done a lot of work, and there's been a lot of history in many other models trying to get to the point that we can simulate many cells and complex geometries and understand that influence on trajectory. There are two key questions of, one, it's great that we can do one simulation with 580 million cells, but do we really, is one simulation actually enough? I think you guys would all appreciate the idea of, you have the cancer cell, you're looking at its trajectory through many different red blood cells. And one simulation is very, if we're doing this right, it's a deterministic simulation. Every time you run it, it's going to be exactly the same. Is that really capturing how the cell is actually moving through the body? We're not capturing any of the uncertainty quantification with that one, directed simulation. We're not capturing, we don't have the imaging capability to say, what we're trying to replicate. If you're trying to simulate 580 million red blood cells, we don't know what the orientation is of all of like 580 million red blood cells in the geometry you're trying to look at. And those orientations can significantly affect the trajectory of these cells. So we want to be able to understand how many cells or how many different red blood cell distributions do you actually need to simulate to capture that uncertainty and understand the effect of the red blood cell distribution on the cells. And it's great that we're able to get down to individual ligand receptor pairings. We can do this in parallel, and that's fantastic. But the simulation that we're looking at, this is still only about 570 microns across. So how do we actually push that and capture cellular interaction at a much larger scale? So try to go through this. We have one project where Cheyenne, one of our graduate students, is leading this. The first step was just how do we even quantify the differences in the distributions? And that's where we've been actually working with Eric Drager at Lawrence Livermore, trying to understand the best way to say, if we want to sample many different distributions of these red blood cells, how do we even pick what the distributions are? How do we determine that the distributions are different from each other? How do we describe these different distributions? And how do we ensure that we have created these distributions and not embedded any accidental structure in these, in the way that we've even just created these underlying red blood cell distributions? So we've started out trying to set up a list of different types of metrics to compare them, and we're currently building the heuristic of, now that we know that we can quantify the different red blood cell distributions, how do you determine all the number of configurations you actually need to run and ensure that you have effectively sampled that distribution space of the ensemble of potential red blood cell distributions to ensure that you're getting strong data and able to make the analysis. And again, I think this is a great place where any kind of the data science techniques are ripe for applying in this area. I know I have like 5 minutes. I'll try to go through this. Our other grand challenge that we've been looking at is how do you get those cellular interactions, but do that over a centimeter scale? It would be amazing if you could capture all the red blood cells across the entire body and get all of the cellular interactions on the scale of the whole body. that's not, not really feasible from a brute force method. I think this is, to me, this was, it helped explain that concept of, this was running on all of Summit, the big supercomputer at Oak Ridge. We were able to get 550 million red blood cells by using the entire system at Summit. And that was about a 3.1 millimeter diameter, or 3.1 tall, little tiny cylinder region. Which is great and a good, it moves us forward, but really doesn't get us where we want to be. We want to be able to capture the fluid movement and capture those cellular interactions over long periods of time. So one thing we've been developing is what we're calling the adaptive physics method, refinement method. It's very similar and kind of built off this idea of like, similar to an adaptive mesh refinement, but we actually have different physics happening within what we call our little moving window and outside the window. Basically, we have this high resolution window around the cancer cell. Inside that window, you're capturing the individual cellular interactions. Outside the window, outside that window, you have the bulk fluid, so you're able to identify, are there vortices? What is the wall shear stress? The overall bulk fluid that we may need to know to set up the overall environment. With that kind of hypothesis that, the red blood cell interactions 2 centimeters away may not actually be influencing where that specific cancer cell is going to be moving, but the overall fluid and the interaction and the fluid behavior at that point is. So we're trying to find methods and develop ways of coupling all of this. And I will say on the data side and just the data scope side that I think was like an interesting side point, this picture, which was actually made from Liam Krause, made this from Livermore as well. We also had like interesting like side problems that I won't really have time to go into here. But just trying to create this image, we ran the simulation at Oak Ridge and we were trying to get the data to Liam at Livermore. And just the shell of that box was about, I think it was like 450 gigabytes. Just to get the shell of all the red blood cells. So kind of just, you have many other issues of like just the scope of the data and the size of the data is getting incredibly difficult. So we're also working with a team at Argonne to create in situ analysis capabilities and situ visualization capabilities and things like, because it's, we're hitting that point where it's just not tractable to actually output everything and do the post hoc analysis. I want to point out that like this, this APR method is actually, we've shown against EFSI, which is really just a brute force model of explicit fluid structure interaction modeling. You can actually recover the cellular interaction, cellular behavior by taking this approach, which I think is really exciting and shows how we can move and kind of bridge the centimeter scale and kind of opens up a brand new area of questions. I really like this example of where we have the expanded channel because it gives the idea of, if you were running a simulation just in the box without the coupled area to the rest of the geometry, you would not see the fact that there is an expansion if you're looking at this box here. So you wouldn't have the margination that's going to be caused by the presence of the expansion. So we need that bulk fluid. We need to understand the setting that the box is in and have that happening accurately in order to get the real behavior of what the cell is going to do as it moves through that geometry. But by coupling this, it allows us to get to this cell-cell scale interaction over the centimeter length scale. So it was really exciting from a data standpoint. The explicit fluid structure interaction model in this bifurcated channel required about 22.5 terabytes of data to simulate. But as we moved down to using the APR method, we're actually able to do it in just 216 gigabytes, which from our end makes it incredibly transformative of the types of questions we can answer, because you can do things like actually, start running simulations that are going to take a week or a month or however long. We were never, at least I would never be allowed to have Summit for more than, 12 hours or a day or two. I would never be able to run a full scale Summit simulation for maybe an entire month or, even an entire week that you might need to get the trajectory of the cancer cell to move a longer distance. But when you've moved the model into something that only requires 216 gigabytes, it's suddenly more tractable to get that onto a node that you can actually run for longer periods of time and actually get longer trajectories. So it's really changing the kinds of questions that we can actually ask. So with that, kind of going into what's next and what are we thinking about and where can we bring in the data science questions? As I mentioned with the moving windows question of, it was 450 gigabytes just for the shell. These the simulations we're creating are really using the full memory of the full system that we're running. We are creating petabytes of data per time step. I think that's exciting and amazing, and that we can, it's providing tons of data that you can feed into these machine learning models. It's tons of data that there's opportunity for training these models and using this amount of data in a way that we haven't been able to yet. But we need to develop ways to actually access that data, and how do we actually use it. So we're doing a lot to set up the capability of accessing this data while it's in memory for the simulation, whether that be for machine learning training or for visualization. But we need that, kind of getting those hooks in place. But then it'd be great to work with people to figure out, like, now that we can access the data, what can we actually do with it, and how do we take that to the next step? I won't get into it here, but we also, portability has become a huge issue for us. As all of these new supercomputers are kind of coming out, and we're working with all of these teams, we're trying to run on Aurora, which is going to be the Intel side, right? So we have, I should add it now, like as of this week, we now have a HIP version of the code. Like we have, the code is running on every single different version. So and there is a question on here, and it kind of fits in what we're doing here of like, are we using cloud resources and how are we using them? And I think that's a great question that kind of feeds out of what we just came of. Like, the work we're doing with like the APR model has now moved it where you don't need all of Summit to run it. You can actually run it in the cloud. So the newer models we're running with red blood cells in the window, and we were actually trying to track cancer cells surrounded by red blood cells over centimeter distances, and we're able to run those models in the cloud. That allows us to run it much longer. So we are working with both AWS and Microsoft to see when can we move data to the cloud, how do we do that? We're also running a lot of the cardiovascular simulations and trying to see, how do we get that running in the cloud? Because as you move to the clinic, the cloud is a great option. How do we approach that? So that's another thing that we're, as we're looking forward to the computational side, we're really interested in how do we make this useful in the clinic, and how do we make use of, not just leadership class systems that we might have on the DOE, but how do we make it more available and useful to other people? And then I'll end with, I think, the kind of broad term of where we're looking in the future. We're really excited about moving from a single heartbeat to longitudinal maps, meaning, when someone is out and basically out in the wild, leaves the doctor's office, can we actually predict what your hemodynamic map is going to be, not just in that one heartbeat, which is really the state of the art today, but can we build a map that's saying this is what your hemodynamic, what your hemodynamics look like over a week, over a month, maybe even over a year, and say not only is, wall shear stress under 2 pascals is, maybe a risk zone, what percentage of the time are you spending in that risk zone? How does that affect our ability to determine what's the best treatment for you? And even can we start giving you advice of maybe you should be exercising half an hour or an hour? Make these really personalized predictions that are going to impact longer term view of the hemodynamics. And to do that, we're starting to do a lot of research coupling it with wearables and seeing, can we drive these simulations from wearables, and create have personalized models from your personalized images, and then driven from data that we're getting from wearables. So we can actually see, for the first time, as people are going about their daily lives, we're getting that continuous data, that continuous monitoring, and seeing, how is your blood flow reacting, and how are you responding to your day-to-day activities. And again, from the data science, capability, as we're launching a 200 patient trial in the summer, where we will have, each of these patients will be wearing the wearables. We'll be running imaging and trying to get their simulations to kind of capture longer periods of time. We are now in that position where we believe our simulations. We've kind of spent the last 7 to 8 years creating a validated model that you should trust. And the big question now is, what can we do with it? Where can we go? And how do we fit in these extended models, like, more intelligent, more advanced data science techniques, and how would you bring that into these longitudinal maps? How do we analyze them, and what can we do with this data? So I'll stop there for questions and say, obviously, this takes a huge team to put together. We've had fantastic collaborators and students over the last few years, and I'd love to answer questions and see what your thoughts are.
Thank you, Amanda. That was a very incredible body of work. It's very exciting to see that somebody is working on such important tasks and the potential to really impact us going forward. That's incredible. We do have, and we're over time, but if Amanda has a couple more minutes to stay on for questions, maybe we could field a quick 1 or 2 questions from folks in the audience. You can either come off mute or type it in the question in the chat. I do have a question for you, Amanda. Admittedly, this is nowhere near my area, and you probably covered this, but this is new information for me. You talked about this synthetic library that you were building up of different simulations, and then also this, the overall goal of having this personalized uh, the predictions. How do you take a body of information that's not my personal geometry, but some other geometries, and then help map that onto make inference about my personal situation?
Yeah, so and that's yeah, that's very useful. I think the way we're trying to build up the synthetic database is trying to understand, are there guiding principles like for the bifurcation project of you know, how do we determine, is it the bifurcation, like, what is it when we look at the large population of like, what are the, like, what is the influence of like the length of the lesion, the severity of the lesion on things like your fractional flow reserve or your wall shear stress? And we like, by having the synthetic database that we've kind of created systematically, we're able to change the stenosis and kind of see how that affects. And we can kind of we can get at what those synergistic relationships are easier, like by using this large scale synthetic database and identify these relationships that we can then go and apply to your specific case. So we can get like this decision tree built up and saying like, okay, we know that the number of side branches are important. And if you have, 3 side branches, that may be a red flag. We can then go look at your specific case and say, well, you have 3 side branches, so we really should be running like this 3D flow simulation. Whereas, maybe if you have 3 side branches, but they're all of a diameter below a certain degree, and the bifurcation angle is something, we can classify that, like use it to kind of drive classification of these of the anatomy and see what may be important and how do we determine, do we even need to run a simulation? Can we classify you as at risk or not at risk without even running a simulation based on our knowledge from the larger population? Or do we need to go and get more information that's really tuned to that patient?
Okay, that makes a lot of sense. Thanks, Amanda. We've got one question from Robert in the chat. Do you have any plans to couple cardiac simulations to your cardiovascular simulations?
Yeah, so I would say like we don't at the moment, but it's something that would be amazing. It's something we've talked about and thought about for a long time. So even like when I was back as an intern at Livermore, I worked with like Dave Richards and Jim Glosley when they were working on the cardioid project back in like 2010. And at the time I was like, I think that it would be great to tie in projects like that with our work. There are a lot of steps that need to happen first on our end that we're kind of slowly taking those steps to get there. Like right now, everything I showed today has rigid walls. So we're not really adapting to the movement of the entire heart as it's beating. It's all coronary arteries on top. We refer to it as the heart model, but it's really the coronary on top of the heart. Like we're not actually modeling any ventricles. We're not doing anything on that end. So I think it's something. What we're currently adding in flexible walls. We have flexible cells, but we're adding in the flexible walls, which should be in by the summer. And we're getting the capability in place that we would need before you would ever even think about adding in like the electrophysiology side of it. But I think it's something that would be really interesting.
Hi, I'm Rob. I asked the question. I'm currently the head of the cardioid project. If it's any consolation, the base of our hearts are also fixed rigid walls. So.
Oh, good. No, I would love. I don't know. I haven't followed as much to know exactly where it's been lately. So it's something of like, yeah, I would love to see if there's a way to connect back to that project at some point in life.
All right. Well, thanks, Amanda, for this very interesting work. If you would like to connect with Amanda and continue any conversation with her after this talk, feel free to reach out to her directly, or I'm happy to connect you to help identify points of collaboration.